This method generates large quantities of 3D hepatospheres from human pluripotent stem cells using defined materials and cell self-assembly.
The main advance of this technique is that it allows controlling the size of the 3D liver spheres, limiting the formation of necrotic centers and loss of phenotype.
Demonstrating the procedure with me will be Wang, a postdoc from our laboratory.
Prepare 2% agarose molds by dissolving 2 grams of low melting temperature agarose in 100 milliliters of sterilized distilled water. Carefully heat the mixture in the microwave with interval shaking to dissolve the agarose completely. Add 520 microliters of the melted agarose to a 256 well format mold and allow it to solidify. Then transfer each agarose microplate into a single well of a 12 well plate. Add 1.5 milliliters of DPBS with calcium and magnesium to each well and pipette up and down to remove bubbles. To prepare the cell suspension, start by aspirating the medium from an undifferentiated culture of human pluripotent stem cells, or HPSCs. Rinse the cells by adding 5 milliliters of room temperature DPBS, calcium or magnesium free, and then remove the buffer. Add 5 milliliters of cell dissociation reagent to the cells and incubate at 37 degrees Celsius for 6 to 8 minutes. Observe cell detachment under a microscope and extend the incubation for an extra 1 or 2 minutes if required. Stop the reaction by removing the cell dissociation reagent and adding 5 milliliters of fresh MTSER1 medium supplemented with 10 micromolar ROCK inhibitor Y27.